This is question number 7 from the same paper, May June 23, paper 22. DNA carries genetic information in the form of genes. The paragraph below describes the structure of DNA molecule. Complete the paragraph by writing the most appropriate word or letter in each space. DNA contains the chemical element carbon, nitrogen, oxygen, phosphorus, and nitrogen. Nitrogen is already there. Carbon nahi. So nitrogen. Okay. In nitrogenous bases, nitrogen is the very important element. Two strands of DNA coil together. Two strands of DNA coil together to form a double helix. You know, one is this, and the helix is this. So this is a double helix structure, double stranded helical structure. Each strand is made up of a chain of nucleotides. So actually each strand is a polymer of nucleotides, and nucleotides are made up of phosphate, deoxyribose sugar, and one of the nitrogenous base out of 4. Bonds between pairs of bases hold the strand together. So these strands are held together by bonds, and these bonds are hydrogen bonds, but this is not mentioned in our syllabus. These bases always pair up in the same way, T with A and G with C. So that was very easy question. Explain how genetic modification can be used to change the function of an enzyme so that it can act on different substrate, or it acts on a different substrate. So remember that enzymes have a specific active site where only certain molecule can bind, which are complementary to the shape. So, and the shape of the enzyme is due to the specific number of amino acid in the protein. So how can we change the shape of the enzyme? Actually, they are asking about this. Explain how genetic modification can be used to change the function of an enzyme so that it acts on a different substrate. So for acting on different substrate, if its shape is this, so its shape should be changed. For example, now its shape is like this, so that another substrate which is different in shape, it can be used. So for changing the shape of the active site, we need to change of the amino acid in this protein, enzymes of protein. So you know that genetic modification occurs due to the deletion and insertion of the gene or changing in the nucleotide sequence of the DNA that ultimately result in the changed RNA and then protein. You know, the general phenomenon is that the DNA, that is the double-stranded helical structure, it transcribed to produce mRNA, and mRNA is translated to produce certain protein, and certain codes are there. So now see how can we incorporate these information that we have studied uh, during our classes should be incorporated here. So we can start our answer from either from the term enzyme or from the term genetic modification. So see, I'm starting from enzyme, that enzymes are proteins while proteins are gene expression. And proteins are the gene expression. So if proteins are gene expression, then we should incorporate other important things from the gene. So enzymes are protein while proteins are gene expression, which are synthesized by polymerizing or adding amino acids on ribosomes using mRNA as a template. mRNA has codes for specific amino acids. While mRNA is synthesized from the sequence of nucleotide on gene, that is DNA. So if we want to change the function of enzyme, function of enzyme, then the change in nucleotide sequence is required, is carried out. So this is very important. Thus, the change in nucleotide sequence produces a different RNA and ultimately for protein, and ultimately for different protein or enzyme that has active site complementary to some other substance. So that's the complete answer for this question that will give you full marks.